Hello students, we are going to study about mycology. What is mycology? It is the study of fungi. What is mycosis? It is the study of diseases caused by fungi. These fungi, they are eukaryotic, non-motile. Their significance is seen in food industry, alcohol industry, in preparation of medicines and vaccines. The various characteristics of fungi, they are usually found in air, water, soil, and plants. Most of them are harmless commensals. They can be pathogenic when they come in contact with the host. They are well differentiated from bacteria, which are prokaryotes. These eukaryotic fungi, they have the cell content which contains chitin, glucan, and mannan. They have a nucleus, various cell organelles in the cytoplasm. They are dimorphic, and the nucleus, which is not present in the bacteria, no cell organelles, and the cell wall of bacteria contains peptidoglycan, muramic acid, and picoic acid. It can be divided into yeast, which are unicellular, molds, which are multicellular, and the dimorphic fungi, which shows both the yeast and the mold form. The classification of hyphae can be based on septate, non-septate, or they can be branched and unbranched. The molds are composed of branching filaments. They form the mycelium. The dimorphic fungi are the fungi which grows in mold and yeast form under different environmental conditions. Most of them, they cause the systemic diseases. The cell structure, the capsule is present in the Cryptococcus neoformans, which is the form which contains the capsule. It is seen demonstrated by the India ink preparation. The cell wall, it is antigenic, multi-layered, made up of polysaccharides, while the cell membrane, it is bilayered, made up of phospholipids and sterols, that is ergosterol and zygosterol. The cytoplasm contains various cell organelles like the nucleus, mitochondria, vacuoles, and endoplasmic reticulum. This is the diagram showing about the hyphae, septate, and non-septate. Then the branched hyphae forming a web, that is mycelium. The budding these cells is seen along with the pseudo-hyphae. These can be differentiated with the Dalmau technique. Reproduction of the fungi can be by budding, the fission, asexual spores like arthrospore, blastospores, chlamydospores, conidia, which can be microconidia or macroconidia. And sexually, they are classified into zygomycetes, ascomycetes, basidiomycetes. Coming to the lab diagnosis, the various samples which are collected for processing of fungi. It can be the skin scraping, the hair follicle, the nail scraping for the cutaneous infections. Then various swabs can be collected, or for deep mycosis to find out the fungi. You can collect the pus, urine sample, CSF, and other body fluids. The processing is done by microscopy, in which the various stains like Gram stain, Giemsa stain, first the KOH mount is done and fast stain can be done. The culture used for the culture media used for fungi is Saprotextrus agar. You can also use the Emmons modification of Saprotextrus agar, malt agar, corn meal agar, bird seed agar can be used for culture of fungi. The Dalmau technique, which is used to differentiate between the pseudohyphae and hyphae. A loop of the test organism is stored on the cornmeal agar and a cover slip is placed on the area stored. After 48 hours incubation, the plate is observed under the microscope. The germ tube test, the single colony of Candida is taken and mixed with 0.5 ml of horse serum or other serum in the test tube and incubated at 37 degrees Celsius for 2 to 4 hours and examined under the microscope. C for the parent cell to differentiate from the pseudogerm tube. The various serological tests can be done to identify the various fungi, pathogenic ones, and antibiotic sensitivity testing can be done by disk diffusion method or e-test can also be used. The predisposing factors for the fungal infection are the immunocompromised patients, the prosthetic devices used in the patients, patients on broad spectrum antibiotics, and ICU patients. The antifungal drugs which can be used are choline derivatives, azoles, allylamines, rhizophlavin, sodines. These fungi, the diseases are classified into superficial cutaneous mycosis. They usually appear on the surface and cutaneous regions. They can be on skin, hair, nail. They can range from mild, but they are chronic. The deep mycosis includes the subcutaneous mycosis and systemic mycosis. They are caused by soil saprophytes. They can be symptomatic to fatal disease. The other mycoses are opportunistic mycosis, mycotoxicosis, and mycetosis. The dermatophytes. Now, what is the difference between dermatophytosis and dermatomycosis? Dermatophytosis, it is the ringworm disease of the nails, hair, or stratum corneum of the skin caused by fungi called dermatophytes. While dermatomycosis is the more general name for any skin disease caused by a fungus. The invasion of the cutaneous tissues by other fungi. The various species which cause the dermatophytosis are Microsporum, Epidermophyton, or Trichophyton, which are based on skin, nail, or hair infections. They are also classified to anatomic locations involved. They can be Tinea bargum, Tinea angium, Tinea capitis, Tinea cruris, Tinea pedis, Tinea magnum, onychomycosis. The ID reaction, it can be lichen staphylococcus-like or Pompolex-like. The lab diagnosis for dermatophytes is with LAMPS examination, direct examination of the scrapings, fungal culture, hair perforation test can be done, serological tests, or animal pathogenicity. For superficial mycosis, the various diseases are the pityriasis versicolor, Tinea nigra, black petra, white petra. These infections, they are limited to the outermost layer of skin and hair. The various etiological agents are Malassezia furfur, Exophiala wernicki, P. horte, Trichosporum bigeli. The Malassezia furfur, it causes hypopigmentation on chest, back, neck area. To identify, it is seen as spaghetti and meatball appearance in the skin scrapings. While Exophiala wernicki, these are two-celled oval yeast seen in the skin scraping. The black nodule of the hair shaft causes the black pigment, which is caused by P. horte, while the white nodules on the hair shaft is caused by Trichosporum bigeli. Coming to the subcutaneous mycosis, these penetrate the epidermis and the dermis to infect the deeper tissues. These are the fungal infections of deeper layers of the skin than those of the superficial mycosis. The treatment, it can be treated with oral administration of azoles. The various subcutaneous mycosis are sporotrichosis, caused by Sporothrix-Schenckii and chromoblastomycosis. The 5 genera of dimorphic fungi are Histoplasma capsulatum, Coccidioides immitis, Blastomyces dermatitidis, Paracoccidioides brasiliensis. These cause the systemic infections, while Sporothrix-Schenckii, it is associated with the subcutaneous mycosis. In deep mycosis, it can be the subcutaneous mycosis caused by mycotic mycetoma, chromoblastomycosis, sporotrichosis, and rhinosporidiosis. These are the diseases which are included in the subcutaneous mycosis. While cryptococcosis, blastomycosis, coccidioidomycosis, and histoplasmosis caused by the dimorphic fungi are included in the systemic mycosis. The opportunistic mycosis can be caused by Aspergillus, Penicillium, Mycor, or Candida. And the diseases caused are Aspergillosis, Candidiasis, mucormycosis, and cryptococcosis. They are usually found in the immunocompromised host, and the portal of entry can be the various routes. While the systemic mycosis is caused by 2 pathogenic fungi, the histoplasmosis, blastomycosis, paracoccidioidomycosis, and coccidioidomycosis. That's it. Thank you.